This video tutorial is all about PCR, the polymerase chain reaction. Modern DNA profiling can obtain results from as few as 5 cells. In theory, if a crime suspect had touched a surface and left a few cells behind, his or her DNA profile could be obtained. However, as we all leave skin cells behind when we touch a surface, a frequently touched surface could transfer innocent people's DNA onto the hands of a criminal, and from there onto a crime scene. In 1983, a guy called Carey developed a technique known as the polymerase chain reaction. Now, this PCR process was used to amplify, so increase the amount of DNA, enabling it to be analysed. The PCR soon became incorporated into forensic DNA analysis and into the protocols of analysing DNA for genetic diseases. The PCR is artificial replication of DNA. It relies on the fact that DNA is made up of 2 anti-parallel backbone strands, that each strand of DNA has a 5' end and a 3' end, that DNA grows only from the 3' end, and that base pairs pair up according to complementary base pairing rules, A with T, C with G. The PCR differs from DNA replication in that only short sequences of up to 10,000 base pairs of DNA can be replicated, not entire chromosomes. It requires the addition of a primer molecule to make the process start, and it requires a cycle of heating and cooling to separate the DNA strands, bind the primers to the strands, and for the DNA strands to be replicated. So the PCR is a cyclical reaction. At first, the process was quite time-consuming as the DNA was heated to denature in it, and then cooled to around 35 degrees C to anneal the primers and allow DNA polymerase to work. Later, DNA polymerase was obtained from the thermophilic bacterium Thermophilus aquaticus, and the enzyme is called Taq polymerase and is stable at high temperatures. So the steps are as follows. The sample of DNA is mixed with DNA nucleotides, primers, magnesium ions, and the enzyme Taq DNA polymerase. The mixture is then heated to around 94 to 96 degrees C to break the hydrogen bond between the complementary nucleotide base pairs and thus denature the double-stranded DNA into 2 single strands of DNA. The mixture is then cooled to around 68 degrees C so that the primers can anneal, that means bind by hydrogen bonding, to one end of each of the single strands of DNA. This gives a small section of double-stranded DNA at the end of each single-stranded molecule. The Taq DNA polymerase enzyme then can now bind to the end where there is this double-stranded DNA section. The Taq polymerase is obtained from a bacterium that lives at high temperatures of around 72 degrees C, which is the optimum temperature for this enzyme to work. The temperature is raised to 72 degrees C, which keeps the DNA as single strands. The Taq DNA polymerase catalyzes the addition of DNA nucleotides to the single-stranded DNA molecules, starting at the end with the primer and proceeding in the 5' to 3' direction. When the Taq DNA polymerase reaches the end of the DNA molecule, then a new double strand of DNA has been generated. The whole process begins again and is repeated for many cycles. The amount of DNA increases exponentially. So you start with 1 section of double-stranded DNA, which will then become 2 sections of double-stranded DNA, which then become 4, become 8, become 16, 32, 64, 128, and so on.